3. Thank you for coming out. And don't worry, the microphone is working. It's for our friends that are we're live streaming too. So don't worry. Yes, I figured I was loud enough already. Well, thank you all so much for coming. If you're new to REEF, welcome. If you're returning to REEF, welcome back. We're so glad you're here. This is Fish and Friends. We have this social and lecture series the second Tuesday of each month. And it really covers a variety of topics, everything from marine protected areas with Dr. Bohnsack all the way to plastic pollution to intro to underwater photography. Definitely check the schedule and see what's going on each month. It's an incredible community to meet, and we'd love to see it back. As far as community announcements go, we have our book club that's virtual and it's coming up on Thursday the 19th. Is that correct, Katie? I feel like that's Thursday the 19th. So definitely check that out. You can register online on our website. And before we get started, I'd also like to introduce our new group of interns that we just got at REEF to kick off the REEF. So come on up front. We're very excited. REEF hosts a cohort of marine conservation interns each semester, young professionals that are kicking it off in the field. And so we'll start on the far side with Madison, who is here from, where are you from, Madison?
I'm from Illinois, about an hour south of Chicago.
Excellent. And Emma?
I'm Emma from Minnesota.
All right. Ben, where are you from?
And I'm Ben, outside of Boston.
All right. In addition to hosting 3 marine conservation interns, they all dressed in our old trip shirts today. So excellent. Color-coded. Perfect. And we also have 3 marine conservation fellows that are here for a whole year. And they focus on a specific program at REEF, anything from education outreach to our invasive species program to communications. So Alexis.
Yes, hi, I'm Alexis. I'm the education and outreach fellow. And unfortunately, we did not coordinate our office.
I'm Dylan. I'm the Conservation Science Fellow, and I'm from Oregon.
And I'm Katie, and I'm the Communications and Engagement Fellow, and I'm from New Bern, North Carolina.
Oh, New Bern.
All right, excellent. Welcome to REEF, y'all. We're so excited to see you. All right,
You don't
perfect.
go in the back. You got to sit in front. There will be a quiz. There's going to be a quiz.
All right. Well, we could not be more excited to introduce Dr. Jim Bohnsack, who is REEF member number 7. And just to let you know how many REEF members there are now, we are just past 81,000. So Jim has been here since the beginning.
Yeah.
And without further ado, Dr. Jim Bohnsack, thank you so much for coming today.
So I had to say, as one of the founding members, when I was asked to do this to help with the science aspects of the program and sampling. So I used to introduce myself as James Bohnsack, 007, licensed to count. So anyway, so that was my REEF background, and I had the pleasure for many years to work with that. So my background, I have a PhD at the University of Miami, went to Tulane University undergraduate. I was a Navy officer in Vietnam. Given my age, I just realized I got first certified to scuba dive in 1963. So that's 60 years now. So I'm still kicking and doing it. So anyway, that's, background there. And for the last 40 years, I've been working in the Florida Keys, which I'm going to talk about today, changes in the Florida Keys. In 1997, the Florida Keys National Marine Sanctuary established the oldest network of marine reserves in the United States. That was the really first ones. California's gone way past that since then, but that was the first one. It was very controversial. People around here back then, you talk about marine reserve. What is a marine reserve? It's an area there's no fishing or no extraction unless you have a scientific or education permit to do so. And so it's no take, and try to explain to fishermen. My role in that, I was a fisheries scientist. I worked for NOAA for about 40 years, or 38 and 3 years in the Navy. And so when I retired in 2019, I ended my career there. So I now live in North Georgia, but still, this is the research I did for about 35 years. So talk a little bit about it and get some high points to you. This picture, first quiz, what's the fish on the left?
No, it's a hybrid.
Townsend. Who said Townsend? You got it. Yeah, so it's a hybrid between a blue and a queen. You notice the ocellate's not quite right up here. It's got the yellow tail. So you know your fish. Fish are the characters in the play. And these were pictures I took, and this is when I took of my catch. When I first started, I was a fisheries scientist, worked on fisheries, and this is a typical fish catch going off Almirada back in the 60s. So one of the problems we have is balancing fishery use and conservation. And so I'm going to talk about the major threats in coral reefs. Let's see how this works. So here's the Florida Keys. It has a rich tapestry of different zones for all kinds of purposes. The reserves are all these little 23 different things here, but we have fish and wildlife areas. We have national parks, Biscayne National Park, Everglades National Park, Dry Tortugas National Park. This is the boundary of the Florida Keys Sanctuary. We have 5 different agencies managing fisheries. So it's a real complex. How do we sort this out? So we simplified it. Either have no fishing or fishing. And so that's what we'll be talking about. The threats to reefs are real and they're major. First of all, in the historically, the biggest threats to coral reefs is fishing. All over the world, fishing is the #1 threat to most fisheries. And the risks are depletion of the species you're after. These are all yellowfin tuna speared illegally in the Bahamas. But depletion of the species you want for your fisheries is an issue. This is a net that was discarded carelessly in the Tortugas, and you see how it's damaged coral here. Habitat destruction from anchoring or line tangling in the corals, just bad practices. Habitat damage, and probably the one of the most difficult for us is bycatch mortality of target species, non-target species. So this is actually bycatch. This is shrimp fishery where they're throwing over. These are all what's left over after they take the shrimp out. And for every pound of shrimp, there used to be about 10 pounds thrown overboard. But these are all little fish. The red snapper here all have commercial value. To some extent. So that's bycatch is one of the ones we deal with. That's a real problem. The third one, well, let me go back. I a little bit too fast. Hang on. So that's the fishery side. We also have a growing human population, South Florida. When I came in 1962, there were 5 million people in the entire state of Florida, here somewhere. And now we're at 21 million. 5.6 million just live in this area between, Dade, Broward to Palm Beach. I mean, so that's a third of Florida's population, very close to our reefs. And so people like to go out and fish and enjoy them. We have a growing recreational diving industry here. It's probably as valuable as a fishery. We have changes in hydrology and pollution, land-based sources of pollution. But we also have environmental threats from climate change, including hurricanes, more frequent, more intensity. We have increasing trends in water temperature. The water is becoming more acid, which is particularly difficult for corals. But also we have things with invasive species, lionfish, and reefs made a major role in terms of dealing with the lionfish as invasive species. So these are the major threats, but I also want to say there's a human threat. And this we call this shifting baseline syndrome. And that is people get, when you set your baseline based on your experience. For interns who go out in the reef, this is great. Look at this, man, all this fish here. I started in 1963. It ain't the same to me. My baseline was established a long time ago. So each generation, the trouble with the shifting baseline, as things deteriorate, people start accepting it. They don't realize how could it be, how good it could be, how bad it could get, and we need to do something to try to maintain that. One way to do that is marine reserves, where we have known fishing, so it gives us some chance to see what it's like without fishing. So that helps change the baseline the other way. People expect to see certain things. And for example, when I started, at Lou Key would see one black grouper about every 100 dives. After we did the marine reserves, then 6 years, we'd see about 30% of the dives would have black grouper. That's your baseline that, again, people get accepting it and they accept the lower quality habitat. And the problem then when our managers side resource, how much we want to spend on resource, you don't get public support because people accept the way it is. So it's important. It's one of our threats, I think. I just want to mention that. And this shows you the human population. This is done on Columbus Day. All the boats here in Biscayne Park. And they're doing all kinds of things. They're fishing to who knows what else going on. It's crazy. But this just shows you the population threat because we have that potential here. And REEF makes an important contribution to our knowledge by changing the ability to understand what we're doing, how important it is, the appreciation for our resources, education, understanding, and appreciation of natural resources. That's what you're doing. With the work you're doing out there, you're getting educated yourself, you educate other people the importance of this, and that's an important thing to do. And I really am pleased with how REEF's success in doing this. We need to balance, in this case, recreational use. By the way, this was taken in 1982, Lou Key, the shrimper ran aground, they weren't paying attention. The other picture is fisheries. So balancing fisheries, conservation. These are divers out here. There are people out here doing education. This is a sea camp boat, city students out there. So how do we do that? And one way is marine reserves. So what do you mean by marine reserves? Again, it's a no fishing area. Oh, you decided to join us. Thank you. Okay, they get 3 extra points. Okay. So what do you mean by marine reserves? And the point being here is we now have the technology that we can remove fish faster than nature can replace them. That to us is an obligation now to manage our own influences on that. It means better understanding, appreciation, and awareness of what we can do. And like I say, responsibility, self-sacrifice, and obligation to maintain our resources. That's one of the things that encouraged us to do, and that's what one of REEF's missions is to do. So I have a little cartoon here that explains how marine reserves work, because usually when we start, everything's depleted. So maybe we need to protect it. You should protect it before it gets depleted. It's better to do that than to have to restore everything. So we make some portion of our habitat marine reserves. Our questions are, how big and how many do we need to balance fishing and resource protection? So here's a little cartoon of what a marine reserve does. First of all, fish settling into the reserve can grow, colonize and grow, and get bigger. While fish that go off into the fishing zone, which is fine, get caught. Whoops. Bye. So you protected it. So the first thing is growth. Bigger, older fish produce more and more offspring, and it's exponential. Fish this big, red snapper this big, 4 years old, produces 170,000 eggs. Fish this big produces about 8 million eggs. Yeah, so exponential means the big ones are really important. We need to protect those, and that's what reserves do. So that's one of the things, colonization, growth. We get more abundance over time. Fish accumulate in reserve. Starts looking different than it was before. So now it becomes worth protecting. When you first start, you don't really care because everything looked the same. But now it looks a little different. So that's the second thing, increased abundance of targeted species, but also increased diversity because we have fish that were eliminated because they're really dumb. We have a lot of fish like that, especially the grouper hogfish, probably one of the dumbest. They get in a trap, you spear them, turn around, let you shoot again. And they just kind of turn sideways to you and try to disappear, and that doesn't work well. But anyway, so eventually marine reserves, we have high diversity, but also benefits fisheries. And the way that works is, first of all, by spillover. That's an important word, spillover. There's so many here. There's a lot of food out there, so they don't know the boundaries. They don't read. So they go across the boundaries, and there's more food out there. So some get caught, and that's fine because it helps support the fishery. So the fisheries benefit. Because they say, well, you're taking this away from it. The first thing you talk to fishermen. You're taking this away. No, we're not taking this. Is helping you. You need these things, reproducing enough of them to maintain your fishery populations. And then finally, increase reproduction and dispersal. So more eggs, more larvae. Some return close up and replace the fish in the reserve, but they also go intermediate distances or even far out, hundreds of miles, because the larvae go hundreds of miles. So, by the way, for every million eggs, the chances of survival to settle are maybe 1/2 of 1%, and to reach adulthood once in a lifetime of an adult fish. So, they produce millions of eggs because most of them die. They're eaten, go wrong place, gets too cold, too hot. Jellyfish get them, all kinds of happen. So this is the idea of a marine reserve. And a lot of my career was based proving this, because back there was not a lot of data. So it turns out, now I'm talking about the Keys. So here's the Florida Keys National Marine Sanctuary. These are all the marine reserves named here. The pink here are corals. We actually have 2 domains. We call the Tortugas domain from about here over, about 60%, 65% or so, 70% is up here in the Upper Keys up to Biscayne Park, right there in Miami. So this is our reef track. I'm only focusing on the Florida Keys for this talk, even though we've collected data elsewhere, because that's the oldest one. Things happen to Tortugas are different. They produced larger reserves there, but it was later. And oceanographic, everything's different. The Tortugas, there's nobody there. Here, everybody, this is accessible. Get a boat, go down the highway, watch what happens. I mean, everything's accessible here and close to the land-based source of pollution. Tortugas is more oceanic, so we're looking at only the Florida Keys. This is a typical thing in 1 year of a cruise. We started covering before reserves were established, and these are samples. All these little dots represent 4 dives. All the little red dots. The blue ones are little reserves here. You can see this is the Western Sambo Ecological Reserve. Up here is Pennycamp Park. We have, the larger reserves here, Pennycamp, Curry's Fort, that kind of area. So this is what we would do. We'd take boats out and we'd do this every year for a while. Then around 14 or 15, started doing it every other year because we started moving into the Caribbean to do the U.S. Caribbean. So there's only so much money we have, and we started doing other years. So for 4 years before the reserves were established, 97, we went around. We didn't know where they were going to be. So we went to all the reefs we could find, dove on them, get some baseline data before it. And then in 2007, they established a reserve in July, but there was no enforcement for a year. So 2007 doesn't count. So we had 4 years of baseline data before the reserves so we could measure what the reserves did. If you have if I made something you don't understand, raise your hand or somebody say something. I'll try to do it, but I'll try to cover it. So what we did is different than what most scientists have done. Usually have a reserve, they compare it, and they try to pick areas similar to it that aren't reserves and compare it. We have what we call, that's an experimental design approach. You have several reefs reserves and several reefs that aren't reserves. We do a survey. We do all the reefs. All reef habitat gets potentially included in our surveys. And we stratify, and I want to say what stratify means. It's really important. So it's based on the variance of the data. Now, 80% of your, say, land, we're looking for people. 80% say desert or mountaintops. There are no people living there, so you spend very little effort there. You put all your effort where the people are, in cities, along coastal areas, and that's what we do with the fish. And we don't decide, I don't say, oh, this is a habitat, and that's the habitat fish like. The fish tell us, because we collect the data. The data tell us this is where this fish likes, and this is where another fish likes, and we can actually divide that statistically into habitat strata. So I'm going to show you how we do this. So the way we do this, some of the variables are cross-shelf habitat types, from shallow water to deeper water. That captures depth and a lot of issues there. Relief and patchiness, I'll show you a picture of that. Those are the 2 factors fish care about. How rugose it is and how big is the patch. If you have a small patch, it isn't going to support parrotfish because they have enough food. A big patch can support parrotfish. So size is important, patch size. Depth, it goes with cross shelf, but depth alone is important. Geographical region, closer to Miami versus distance toward the Tortugas. And then we have a human zone. That's fishing allowed, no take reserves, fishing's not allowed. So those are our strata we use. And we, again, put our sampling into that kind of strata. So here's some of the 7 different kinds of reef habitat we get. We have inshore patch reefs, mid-channel patch reefs, offshore patch reefs, reef flat, the 4 reef area, and then the deep reef. Reserves only in the Florida Keys only went down to 18 meters, around 60 feet. And then we're stopped there. So we actually have strata deeper than that. We counted, don't have reserves there, but we have, so we have data from there. So and here's what we did. We take all the habitat. The only way we could do this is have really good maps. So we know where the habitat is, and these are the color coding telling you the difference. This is the mid-channel patch reefs, offshore reefs here, low relief, hard bottom, patchy reefs, spur and groove here. These are colored. So we divided the whole Florida Keys into 200 by 200 meter grids that had coral. Things that don't have coral, sand or seagrass or mud or something isn't included. So these are where all the reefs is. We did it for the whole Florida Keys. Here's the Western Samo Marine Reserves, for example, and you see this reserve has quite a few different habitats. It's more of an ecological reserve because it has different habitats. Some of these are just the 4 reefs. Blue Key started as a 4 reef and got bigger. So this is how we do our sampling. So this is the first stage. We just randomly pick the habitat based on how many we know we need to have a certain number. And each year we collect the data, reanalyze it, so we learn from that. So we modify our sampling design based on what we learn. So, maybe a little bit too much. Whoops, I lost things. So this is the second sample. I'm doing this because reef, it's based on, the roving diver technique is based on how much time you go searching. So you dive in, you search, but you may get different habitats in there. For us, as scientists, we need to control how much area you've sampled. And rover diving go all over the place, and we don't know how far you went. And some people have a list. They know if I go over here, I'll find the lantern bass or something, if I get this kind of rubble habitat or whatever. So they have all kinds of methods. We randomly select areas. It has to have reef habitat. We go down, we send 2 divers down as a buddy team for safety. So I go down, my buddy will pick an area, and I'll be very close in the area. We may overlap somewhat, that's fine. We take our data, average them together, and cut it in half. That's the sample size. And what we do, this is a radius. It's 15 meters, so 7 meters. So it's about the size of this room. I get in the middle of it. I can estimate very good the distance. And so I sit in one spot. The idea of sitting in one spot, fish habituate to you. You swing all around. Some fish dive in the hole. You never see them because I don't like this big thing moving around. But if they habituate to you, it means they get used to you. They also come out and start adding, yellowhead jawfish, for example. If you're moving, you won't see one. Sit still, and all of a sudden they kind of come out of the holes, and a couple grouper come out. So the idea, and this is the way the military does surveillance, by the way, too. They find an area of interest, they put somebody in there, and they just sit there and watch what's happening in the neighborhood. That's the way they do it, sitting still. You get to see what's going on. So that's the way we do our diving. This is what it looks like out here to us. So we would look at, so for 5 minutes, I have a limited time. 5 minutes, I list all the species I could identify from one point. I get 30, 40, just not even moving in that time. Too long, you start accumulating things that are highly mobile, and it biases your data. So again, we have to know the distance. We know that in order for the statistics to work out. So we scan the horizon. I sit here and I'll go around, look around, and just like here, there's, French angel, queen angel, hogfish, blue reef fish. So those are the ways we do it. We have a ruler we carry, a meter stick. So it's a big fish. Let's see, that's shark. Let's see, that's 3 feet. That's, meter, 2 meters, 3 meters, 4 meters, a big hammerhead. I remember one time my knees actually shook. Hammerhead came right over my head. You always hear you get so your knees shook. My knees shook. I was trying to look like a coral head down there best I could. I noticed my handwriting got really big. But normally it's not that exciting. But anyway, then we have I have a ruler. It's in 30 centimeter increments. We do it to centimeters, to the nearest centimeter we can estimate. So the data we collect here. So let's see. We do the next one. Here we go. So here's an aerial view of one of our circular plots. This is Blue Key Reef. These are actually spots we did early on. Here's a cartoon of our cylinder. The diver's in the middle. Kind of looking around. This is my wife who's joined us here, Barb, out here with her stick, back looking at some mackerel going by, trying to estimate their size. And this one is a hogfish. So the idea by being still in that radius, we're optimizing for exploited species. They may not go too close to us, but we can see them, and we can estimate their size. I could even see, something small like a glass goby or something, I can see those. So the data we collect are the species observed. So I have my list. I go back up through the list very systematically. I try to figure out how many there were and the sizes and classes. Sometimes, there'll be a bunch in 15 centimeter tomtate, or a whole bunch of little ones, 3. So we do that. We have how many individuals. Then we can estimate biomass because the size. We figure the biomass, add them up. We use frequency here. How many times do we see them? 30% of the samples, like the most common thing you're going to find is bicolor damselfish, probably, and bluehead wrasse, especially neperkees, bluehead wrasse. And then we collect data on the habitat, depth, relief, and the profile of the habitat. So this is what we do and why it's different, the reef example. So the important thing that we do that's different is it shows that each of these patch reefs has different composition when you do it, how much it is. So the 4 reef deep, kilometers square, there's no reserve in these areas because that's the minimum, the maximum depth is 18 meters of reserves. There's none here, but there's a lot of habitat. So you notice most of the habitat is in the 4 mid-reef area. These are fishing zones. These are in kilometers. Sorry, that's why we wear kilometers, square kilometers. But it turns out, if you add this all up, it turns out reserves only cover 4.4% of all the reef habitat. The other 95.6% are fished areas. So most of the reefs is fishing. And I point out when they did this reserve, it wasn't for fishing. It wasn't fishing. Marine Sanctuary does not do fishery management. But they do have to manage fishing because it can damage habitat. And so the big issues there were habitat damage by fishing and interactions between divers. That was big. So they picked areas that are favored by divers for these marine reserves because it's kind of embarrassing when someone's trolling for barracuda and runs over a snorkel or something. That's not not a very good interaction. So the idea is to make incompatible actions. The other issue is if you're a diver, you want to see fish. And right now, at those times, whatever's left over after the fishermen are done, you got to see. So it wasn't very much there. It's the Black River story I told you. So again, this is the composition. Each of the reefs have different ones in marine reserves. We count for it. And each reserve has its own composition. So here are all your marine reserves. And you can see the different habitats. These are the same thing, like offshore protected area or the various fishery zones. The total here is like 30 square kilometers for marine reserves out of 600, well, there's about 500 total marine kilometers in the Florida Keys. And so about 30 is that. So it's 4.44%. So it's a very small area. So there's no real thought that this would ever do anything for fisheries, but we might learn something from it. So, and this is what we had. It's a political process. It wasn't a scientific process, how these areas were picked. So we do the best we can. So this is the idea. The 2 things that fish really key to is the depth, but also the patchiness. So this is patchiness up here, high patchiness or low. These are very low, flat areas, almost look like, sediment. It's reef, but it's pavement, we call it. It looks like pavement. Or then the high reef terraces are a high vertical relief. And up here, the pinnacles, these came up like 30 meters up, straight up. These are pinnacles. So again, it's high patchiness, high relief. So between these 2 variables, you can explain most fish. It gives a signature how fish use their habitat. So now I'm near, by the way, I had 26 slides. I'm getting near the end here in slides. So, but this is the important part. This shows you what happened. So what we did, we analyzed all the fish. We had, I think, a couple hundred fish identified in our surveys. There's about 70 non-targeted species we had enough data to analyze. There's about 35 target species we can analyze. These are one fisherman target. So over here in this column, actually I have a little pointer here, don't I? Yeah, look at that. So there's 5 exploited species that show significant changes. Yellowtail snapper, mutton snapper, black grouper, red grouper, and hogfish. Red line is what's happening inside reserves. The black line is what's happening in the fishing zones. So you can see immediately they're all very low. All these species, beginning of the study, had very low abundances. They all took off after reserves. The only ones that increased after reserves were fish species. I wonder why. Yeah, because you couldn't catch them anymore. So they settle. Remember the little cartoon I had? They get bigger, they settle, they get more abundant. And these things went through the roof. I mean, it is incredible, the distance. But then, you also notice here, the first year of the thing, we had a little bit of a heat wave, El Nino, but also we had hurricanes in 88 and 89, or 1988, 1999. So that's important here in a minute. I'll talk about that. So these vertical lines are what we call confidence intervals. It tells us how somewhere the true number's in here somewhere. So if they don't overlap, they're significantly different. This is significantly higher than this one. And you can see the only one we had 8 categories of exploited species and reserves and non-reserves. 7 of those increased. The only one that didn't is in fished areas. Yellowtail snapper never increased in fished areas. It's the only one never got significantly above its baseline, but all the others got significantly above the baseline in reserves, but also outside reserves. And that's the idea. It's supposed to help fishing, so there's more chance of catching fish outside the reserves. So those are the 5 species that we found significant differences in. We had 2 species that were fished, but they're under fishery moratorium because of overfishing. And they had significant change, but nothing to do with the reserve. Generally, there's no difference between inside and outside because of the variance here. We did see a drop-off of Nassau grouper after hurricanes in 04 and 05. And Goliath grouper became more abundant after hurricanes in 04 and 05. That's a thought question for you. Why is that? So you can ask me later if you haven't figured it out. So the point being is all of a sudden in 2004 and 5, things changed. We started out looking at reserve effects. That was our goal. Our methods were designed to detect any kind of change. Up, down, we should be able to detect it. Not for all species because you don't have enough data, but these were what we did to detect change. In 04 or 05, we had 8 hurricanes in 18 months. That was the start of a climate change event. Never in history we had that many hurricanes, that quick a frequency. So the first half was reserves. At some point, we started looking at climate change effects. So it turned out we had this major event, and all of the species you see decline. Here, by the way, let me go the other species here, non-target species. These all showed significant changes sometime during the study. So we have the tangs here, ocean surgeon tangs, sharp-nosed puffer, wrasse, yellowhead wrasse, pudding wife, squirrelfish, harlequin bass. I'm just you're fish people, so I want to make sure we know. So we had the parrotfish, the small ones, and large ones. Stoplights showed significant changes, and some of these really went wild. Bright color is probably the most abundant things you see in the Florida Keys reefs. They follow close, inside and outside. The reserves had no effect on them whatsoever. You can see it, but it did have an increase in this point after the hurricanes. This one's interesting. This 3-spot damselfish, and it went down everywhere, both inside and outside. And that's the one that really is closely associated with coral, and coral's been declining for 40 years. So that's their favorite habitat, high relief acropora corals or fire coral. And so they've been declining, it shows corresponding to the habitat loss they're facing. They're not endangered, by the way. They're just much lower possibility. So these are the 3 groups of fisheries. And again, the point being is in 04 or 05, we had all these hurricanes. We see a lot of changes then. Then we have a period after that. It's a response period where there's, where things may come back. Then we had a record cold snap 2010. It killed coral and fish. The coldest water ever recorded here in South Florida in the Keys. And it was definitely death for corals and fish at that point.
How cold?
Depends on where you were. The shallow reefs tend to get colder, offshore less so. And it depends. With fish, they lose control and they start floating, and seagulls get them, or they drift off the wrong area. They drift inshore, it gets even colder. So, but it hit, I think, 13 degrees C. It was cold. It was cold. And then in 2014 and 15, we had the hottest temperatures on weather. We had a 2-year El Nino. We never had a 2-year El Nino before. And they measured a 30-day high temperature, around 31, 32 degrees C. Coral starts bleaching and dying. But also fish, it can affect fish. So anyway, so we have, all of a sudden this year, from here on, climate change type things are predicted by climate change. More bigger storms, extreme weather. By the way, this is an El Nino year, but it was in the winter, extreme cold. And then we had hot. And this is where we started skipping years so we could do the Caribbean. That's why there's a blank here. So this is the data we had. So I just give you an idea how we do it and what it looks like. So I'm going to take this is the important part here. So this is the 5 exploited species. And this is total fecundity. Now, I talked about how they get bigger and more eggs when they get bigger. Well, this is the total fecundity in marine reserves. So for here, for example, you expect 4.4% of, if there's no effect on habitat, of the reproduction come from reserves. However, this is up over half, almost 70% in this 1 year of the total fecundity. The entire Florida Keys came out of those little tiny reserves. 4.4% of the reef is producing most of your fish because they're bigger and older. Big dominates. So you can see up here, yellowtail snapper up to 50% of all the yellow snapper were breeding from these tiny little reserves. They tend to be pretty territorial. Reef fish are. Hogfish, look, this year is almost 80% of all the fish are coming out of reserves. So even though all these were tiny, they really had an effect far beyond any expectation that we had. So that was a good thing. However, notice after the hurricanes, they all dropped. They never came back. And that was interesting because first they went like gangbusters, up here. We went really good. Why didn't they come back after hurricanes? So they're much lower. So it's important to understand. They didn't come back the same way. So that was the question. I'm going to talk about why here in a second. So just a little bit about hurricanes. So it's a natural phenomenon. This is in 1988. We had this is George's, Hurricane George, a Category 2, which is a pretty good-sized hurricane, across Cuba, across the Keys. The green dotted line are tropical storms. They don't do anything. You ignore them, but I just want to put it on. So there was a tropical storm, but the winds are nowhere near a problem here. Then 1999, we had one hurricane. This is Irene. Here are the ones over here, the names. So we had tropical Georges in 88, 89. So one storm in each year. Then in 2004, we had potentially 4 hurricanes. This is a Category 5. By the way, we've learned from other studies about 150 or 250 kilometers away, a fish can sense a hurricane coming. And it's based on the ground swell. You start getting these swells come in, these low swells. The fish feel that. They start going into deeper water. This is done by tagging studies in North Carolina of triggerfish. And so they sense it. 2 different hurricanes, they start going into deeper water to avoid the hurricane. They're adapted hurricanes. Hurricanes kill very few fish, especially adult fish we're looking at to fish. The small ones might get swept away by high currents and things, but very little. But what happens with hurricanes, it homogenizes the population. If you've been out here after a hurricane, it's just mud. Fish can't see. They use cues to vine, so they get lost, and they may end up in reef habitat, may end up somewhere else. So it mixes the outside fished areas population with really high densities in reserves, and the net result is a very low density everywhere. So most of the fish in reserves got moved out. This is spillover, but all at once by the hurricane. So this is why the difference between the two curves here. So let me go back. So here, hurricanes had no effect because the densities were the same inside and out. There's no real density difference, so you just mix the fish. nothing happened. So this may have slowed the recovery, but it didn't have any effect on density. They all stayed low in the hurricanes. So this is, and again, the fecundity is related to their density and size in these reserves. So anyway, so that's what hurricanes do. So it's a spillover effect. The lesson being that small reserves work great when weather is fairly benign. Once those hurricanes start hitting, they all got moved somewhere else between the hurricane damage and then the death from cold snaps and hot water. They never really recovered. That's called resiliency. They didn't recover. It had high resiliency initially, even though they're hurricanes. 4 years, we had significant increases, and some of these were very high increases. But after hurricanes, climate change, no. So that's the hurricane story there. And this is something you probably haven't heard. This just shows you how we analyze it. This is one fish, it's the bicolored amsel. And so we divide into periods. We have a baseline period, it's a 4-year baseline before reserves. We have the post-reserve response period. In this case, it didn't make any difference. This is the average. So if there are difference between fish and marine reserves, we combine the data. It didn't make any difference to the fish. If the only ones that made a difference were the targeted species, those 5 species, so we analyzed them separately. They did distinguish between fished and no fished areas, only the exploited species. So then we had the hurricane disruption here in 04 and 05. We see a decline here. This was significant. These don't overlap. It's significant. Then post-hurricane, they really rebounded. This is high resiliency. You know, they did very well partly because maybe all those fish that used to eat them in the reserves aren't there anymore. Then we had the cold snap, big decline. Post-year cold response period, they increased. And then we had the El Nino towards the end of it and kind of flat type action. So this is how we analyze the data for each species. This is an important part. We'll talk about fisheries. So unlike other studies, not only did we do underwater, we actually looked at fishery landings. How did the fishermen do? What did our fishery landings tell us? So I'm about to tell you that story, but the important point is here's a marine reserve. I think this may be Coffin's Patch. I don't remember. But here's fishermen's boats. This is a reserve. There's a little, can't see it there, but there's a little yellow marker buoy, the quarter buoy. All the fish are fishing the edge. It's called fishing the edge. They figure that fishing will be better. Next to reserve, they're looking for spillover, the fish coming out of the reserve. They may chum them a little bit, try to encourage them to come out. But so the fishermen were obeying these pretty well and complying. The question is, you can't enforce all these, but no, fishermen did a good job. And that's part of this story. So these are fishery landings, and this is a little difficult to explain, but bear with me. So this is a quite different picture. So these are the reserve effects, the 5 target species. These are fishery landings for these 5 target species. So on this side, red, these are commercial landings. They're scaled to their maximum. And this is in different units. This is in metric tons. Recreational fisheries, we count how many fish. They don't do weight. They don't weigh them. They just count how many recreational fish. So those are different units. So we scale this. But you notice after the study, again, 88 is the first year of possible change. We had the hurricanes. Fishery landings declined after the hurricanes initially, both the recreational and commercial. See, they all kind of went down to a low point, and then they increased after reserves to the highest point during the study. So what we did, we divided all of our data into 2 periods. This is the stable period initially. This is the mean landings for that period. Everything's standardized, the mean. This is the mean landings after the reserve period. Remember, it all went down, so it's lower. So this is what happens. For example, black grouper, the average commercial fish in this mean is 36% decline from what it was here. So even though this is scaled to 0, it's much lower. All these show drops, especially recreational fisheries and commercial for red grouper. So we saw a decline, an increase in reserves, decline after hurricanes, some sort of equilibrium. A couple of hogfish and yellowtail snapper, showed some increase, and that may be due to the reserves and the tortugas. At that point, this long distance dispersal was probably benefiting these fish because they're doing very well in the Tortugas. That's not part of our study, but it's a good evidence of that. So I'm missing something here. Oh, the important part is recreational dropped more than commercial. Part of the story there is they all fish next to reserves, or they tended to. They fish anywhere they want, but most recreational fishermen don't know a lot about the area, and so they go out next to reserve. They figure it's good fishing there. So that's the first place to decline, and so they lost their fish, and they showed the big decline. Most of those fish were now out of the reserves in the fishing grounds. So the commercial fish did pretty good for a year. Then they caught them all. It took about an extra year. So that's what happened. So the dispersal failed. So the lesson is small reserves were good on when things were going benign environmentally, but they did not survive or do well under hurricane climate change impacts. So the lesson there, if you're going to do a reserve, you need bigger ones, because when these things get stirred up, they have a better chance of staying in the reserves. And that's another story, and that's the Tortugas. So those are the lessons. So fishery landings tell, a good story complementing what we did here. And again, hurricanes affected fishery landings here and here, even though the densities weren't any different, because outside they were being fished. So this is the results here. So I apologize for, I'll read this to you because I'll make sure I got them all. So if you have any questions, you can ask. So under stable conditions, that's 94 to 2003, exploited species density, total fecundity, and fishery landings increased significantly in the 4 years after marine reserve creation, despite hurricanes in 88 and 89. All recreational fishery landings peaked in 2004. That's before the reserves, the hurricanes. Commercial landings lagged until 5 and 6 because they benefited from the fish being dispersed out of the reserves, supporting their fisheries. But then they declined too. They caught them all. Under variable conditions, again, this is 04 to 16. By the way, this whole period to 16, we ended in 16 for a reason. That's the whole time the fishery regulations were continuous. They didn't change. Now in 17, they started putting size limits on hogfish, for example. That actually improved hogfish very much. And they did a couple other things. So we ended the study in 17. So it went from 94 to 2017 with the same, so you can't say the fishery regulations improve things or whatever. It had nothing to do with it. They're the constant. So that's why we picked those dates. So hurricanes cause mass exploited species spillover from reserves to fishing zones, significant density declines in reserves, and lower fishery landings overall. That was the result of the hurricanes. Recreational landings declined first and were greater proportionally than commercial fisheries because they depended on those reserves as good fishing zones. And the hurricane damage, extreme cold, and El Nino warming impaired the exploited species density, resiliency, and fishery landings recovery. So those are the lessons here that we've learned from 35 years of work. So anyway, so I hope that's, I think that's probably all conclusions. I think I've covered this. Interesting. So initially we had high resiliency to fishing. We stopped fishing, fish took off. The exploited species did really, really well. 7 out of the 8 possibilities increased. So it's 7 increases in 4 years despite hurricanes. Intermediate resiliency, cold snap mortality. We had 3 increases in 4 years after the cold snap. So actually they've come back pretty good after the cold snap. And low resiliency to all the hurricanes in 04 and 05. We had no significant increases in 4 years compared to the first part of the study. And that's because of the loss of the fish to fishing. And it's like milk, it's homogenized. The population got homogenized by the storms and didn't work. So reserves increased fish as predicted. Variable conditions from climate change disrupted problems with fish. High exploited densities and small reserves are vulnerable to mass spillover from hurricane disturbance, mixing density in the populations. And finally, I want to just go back to that thing I said about shifting baselines. That is still the major thing that I don't deal with. I deal with physical things, biological things, but the human dimension is a really important one not to underestimate. People get used to declining resources, and that's a problem. Then they won't support the legislation. Our managers, they based on what people want, and there aren't people saying we want more reserves or this or that. Actually, a good story on this whole thing is that it took 7 years from the creation of marine reserves in 1990 until we got the reserves in 97. 7 years. Actually started before that, but 7 years. We actually expanded the reserves in the Tortugas in 2000. It only took 2.5 years. We had long, strong support because people with their own eyes went out. You didn't need science to tell you what happened. We had people going out. I saw 3 grouper today, and the coral heads off, Big Pine Key. Everybody, they talk it. The dive people saw it. The fishermen knew it and saw it. So when the new ones came and the Tortugas, again, no one lives out there. That helps, but they're much bigger and very productive. And that's another story because they're so different. They're not totally comparable. So those are some of the lessons we've learned. But this is kind of the sanctuary preservation areas, cure for the common dive. So this is extra strength because we have more fish compared to regular where the fishing goes on. Like I say, it's not, bycatch is solved, mortality is solved. There is things you don't have to see what's left over after the fishermen are done. So this is supported by the commercial industry down here in Key West Tourist Commission. So they use this as an advantage in their sales. So that's part of a good story here. And so I'll end with habitat here with mutton snapper. And any questions?
Does anyone have any questions?
Yes, ma'am.
I have a question. Since the regulations changed in 2017, has anybody been doing another study?
Oh yeah, we're trying to do it. Yeah, we did one actually in 18. We did a survey in 18, but we excluded it because that happened. Then COVID hit, so diving kind of got knocked back. But someone who was here tonight, I think, just got off a cruise, right? Was supposed to be here tonight or listening in or something. But so we do that. And by the way, this is a big effort. I started out with all my colleagues there. We all realized when they created the reserves, no agency had the resources to do this sort of thing. So we all got together, different agencies. Say, well, we had the Park Service, we had the State of Florida Fish Commission. I was NOAA, we're at the NOAA. We said the only way we can do it if we all cooperate and work together and pool our resources. And so we did that informally. And so that's the only way it would work. So I had all the co-authors that work in this. It wasn't just me. There's a bunch of us doing it. We all had to compromise, get standardized methods, and do this. So there's over 25,000 dives. I do up to 7 dives a day at one point there. So, but they're short dives. So you go down, you do your count. It go a minimum 5 minutes. You land on sand, it's 5 minutes. You see little pearly razorfish and things. But it could take up to 30 minutes in a really high relief, busy area. And they average maybe 15, 20 minutes, including breaking down the habitat and the depth. Yeah, we had things we wrote down. Yeah, visibility, that sort of thing. So that's 25,000 dives. I think we had about 150 volunteers overall, 149, I believe. These are students, these are faculty members of the university, these are employees from Fisheries Service, my service, the Park Service. We all agreed to hold hands together and jump over and do this. This was difficult. I mean, like Park Service, they only cared about the boundaries. Anything about the boundaries, they didn't care. But, that's not a good, effective strategy. You got to know what's outside. How well are you doing? If you don't know what's happening outside, you don't know how well you're doing, right? So they all agreed. We did it. And this is a major accomplishment, getting this many government agencies work together, I think. So, yes, ma'am.
I have a question.
Loretta, yeah.
Yes. With the coral loss, is there any correlation of the fish population with
Coral loss. Well, you saw the 3-spot damselfish, right?
Yeah.
Definitely.
Yeah.
It had nothing to do with this. I mean, that if you go before that, clearly the habitat was there. That was nominated as dangerous species, but I was one of them say, okay, they're not as frequent. We plot frequency versus abundance and stuff. And they're lower, but they're not endangered at all. There's still a lot of them out there, so, but certainly the habitat. So there is, others are less, there's other things going on in between fisheries, fishery action, bycatch. And I didn't go into it, but we actually found that when the predators declined, a lot of the non-target species increased. So that's another part of the story, and I don't want to make it too complex. There's a lot. I gave you a lot of stuff here to think about. Yes, ma'am.
Going back to your sampling method, when you determined the grids of the benthic habitat, was that ever changed throughout the study? Because we saw all those hurricanes and potential bleaching events, or was that the same sampling sites were classified throughout the whole study?
Well, we did change at one point. We actually went from 200 meters, 2 by 200 meters, to 50 by 50 meters, and you put one sample in it. That's because we improved our maps. So we're constantly testing. So when we go back out, we go in an area and we go down, and this is coral, there's not. We note that. So we try to correct it. But generally, they're fairly stable. I mean, there are some areas on the edges, but so when we go into a cell, we just have to find reef habitat. So, so if there's a little tiny area size of this room and 200 by 200 meters, we got to find this room and go down. So it may be a different reef. And there's so much, we're talking 500 square kilometers of habitat. That's 67% of the entire reef habitat. By the way, the Florida Keys is the world's third largest barrier reef. Think about that. So this is a big area, and most of it is in the Florida Keys. Tortugas is 33%. Again, it's just different. I'm confounded because with climate, and no one lives there, and they've been doing management for a long time. They stopped commercial fishing in '95, for example, in the national park. So it's different, but still, those big reserves did very well. But that's all published, and it's out in the literature. You can go look yourself if you find some help, library or something. But it's done. Well, thank you. You've all been very kind.
All right. Thank you so much for coming, Dr. Bohnsack. We really appreciate you coming to Fishing Friends. Appreciate you. And your early dedication to reef. I know that work is still going on. There's several really active surveyors in this room, as well as people who are really just passionate about Florida Keys and the marine environment around here. And there is actually a REEF alumni, an intern alumni that went on the most recent survey of
That's what I said, yeah.
NOAA. Andrew Ibarra is listening in online tonight. If you have any questions, next Fishing Friends, I'm sure that he's happy to answer how that trip went, because he said it was a blast. So with that, our next presentation will be actually on Valentine's Day, February 14th. So join us Tuesday, February 14th. And Abby Haddock will be talking about, she's from the Dolphin Research Center, and she'll be talking about dolphins and their cognitive abilities and why that's important for us to understand.
Cool.
And with that, thank you all so much for coming. We'll see you next month.
Let me say one last thing about reef. I can't tell you how valuable it was. When I go in front of fishery councils, I call it combat biology. I'm there to tell them, explain the science, and they don't want to hear this, and especially in a chemically altered cloud or something. But so I say, here's the trends. They have somebody from REEF come up with their data saying, yes, this is what happened. We've seen the same pattern. It's collected independently and different than how we do it, but it's really valuable what you do, and keep doing it. It's good. Thank you.